the last three days, which has been very exciting. I'm in a very, very strange position because I'm going to do something I've never done before, give a lecture on Viroids. Even if you believe it or not, I've never given a lesson in Viroids in any course, undergraduate, postgraduate. I've been teaching for 25 years. Accidentally, my first teaching times were in IAMB in Crete, in Chania. I used to teach there for 3 years. There's a branch there. But I've never taught Viroids. So today I have to give a lesson on Viroids, which is a first for me. So I all welcome you here. I've been thinking a lot about what to teach today. And I realized that the title of my talk, which is 50 Years in Viroid Research, will not be super interesting because there are some excellent reviews out there. There is one from Diener, which I highly recommend. There is one from Steger, which I find very, very interesting because it's very different from the others. It doesn't list the event, the historical events, but he goes and states what aspects of viral research have helped generally biological research, which is, I think, a very good start for you. I'm going to somehow, I took inspiration from this review from what I'm going to be talking today. First of all, I would like to say a few things about myself, not because of any importance of this, but because I would like to say that careers are, and especially today, but even in my time, careers are like Lego constructions. They're not linear. You don't have to do one thing. You don't have to start with one thing and finish in that. So I'm an agronomist by studies. I did my degree, my first and second degrees in Prague. So I'm a Czech speaker. And then I moved to biology and did plant developmental biology, which is what I've been teaching the last 30 years. So that's my main, let's say, educational dish. And then I worked on plant biotechnology for quite a few years before joining a lab where viroid biology was very strong, Mina Tsagris and Martin Tabler's lab. Many people here would know them, the older generation. The new, I guess, we will have never heard of them. And there was very active research on viroids. And this also connected me somehow to the people that are here for various reasons, including the fact that Martin and Mina were very active members of the viroid community. Also, Emilio Martinez was at the time finishing his PhD when I was joining the group, then became a connection to Ricardo Flores because he went to do his postdoc there. And if I remember well, it was Emilio. First, Emilio was the person that isolated the protein I showed you in the first day, which was VIRP1, and also Emilio was the first to publish small RNAs for Mafsunoviroid with Ricardo. So there was a connection there. And then after actually Martin Tabler in an accident passed away, I started working with viroids. So I only started working with viroids 20 years ago. But what I would like to show today is how important viroids are and why they have been keeping me very excited about science. And I will try, because as I said, the historical facts you'll find in the reviews. I will give you some historical facts because they're inspiring. But as such, you will find them better in papers than you'll find them in my presentation. But what I can give you, and papers cannot give you, is my inspiration from viroids. So first of all, I would like to all welcome you very much to this ViroiDoc as students. I am very excited that this is happening because maybe I share this with other colleagues, I'm not sure, but I have this constant fear the last 20 years, but not everybody else or not new people will not find this piece of research as fascinating as I have. So I have all this continuously this fear that we're kind of dinosaurs that will disappear. And every time the Viroid meeting keeps reminding me that we are not dinosaurs, or at least there are young dinosaurs around. So there's a hope that at least for one generation we will live. Okay, so I'm very, very happy to see a young generation of people being here with Viroids, and I'll try to keep you somehow inspired about Viroids. Maybe I need the, Thank you. Okay, so that's me. So by the way, we will keep in touch with many of you for various reasons. For Lena, for example, she has to. She's obliged to see me every day. But the rest of you, you will see me. Some of you, because you will have secondments, like Theodora and Judith. And the rest of you, you're very welcome for whatever reason to contact me. Now, unfortunately, the spelling corrector made a mistake here. Everything is small, all small letters. You can contact me whenever you like about anything that has to do with Viroids, also your academic life, whatever you like. You can contact me. Many people contact me every day, so that's okay. I won't take it badly. I have a double affiliation at the University of Crete, where we are also based as a lab, and also at the Institute of Molecular Biology. Maybe at the coffee break I can explain what that means, but it's not time for that now. Okay, so this is stolen, actually borrowed from Ricardo Flores. This is a diagram showing where Viroids stand in the tree of organisms in terms of size. This is a very fascinating slide. It doesn't look in first, but it's really fascinating. Why it's fascinating? Viroids have been isolated not because some molecular biologist one day decided to do some molecular work and accidentally found it, like we would do today with deep sequencing technologies. Today, that's how it would happen. You would stumble upon them. You would do some other analysis, and you found something that molecularly in the analysis of the NGS doesn't stand well. But at the time, this is not at all how it happened. How it happened is that some agronomists, this is important, noticed that plants were diseased. And this disease had to be explained somehow. People for a very long time, for centuries, didn't care so much about the actual biological etiology of diseases for various reasons. The main one being that it was not possible to isolate them. Yeah, they knew that they were contagious, they knew that they were ways of spreading, they knew there were ways of stopping, but they didn't know how to isolate them. And then from the 19th century onwards, especially in the 20th century with the first microbiologists, people started isolating the actual agents that were doing the disease. But as you well know, whatever degree you have done, you know that the disease is not the microbe. Very often you have microbes with no disease, and it can be the same one. Pitch latent is a very nice example. Usually it's latent, and suddenly it's a disease. But it's the same pathogen with a very small change in nucleotide sequence, but it's the same pathogen. So disease is not the pathogen, and pathogen is not disease. We all know that very well. Amongst us, no, not here, although it wouldn't matter, but amongst us there are many people which are now HIV positive but don't have AIDS. Yeah, everybody that has AIDS is HIV positive, but it doesn't mean that the pathogen is the disease. Why I'm saying that? I'm saying that because people were seeing symptoms, and these could not be explained with the usual characteristic of the causing agents of diseases. The first diseases to be identified were microbes, because these were seen. So because they were seen under a microscope, and that's why microscopy became so important, and that's why microscopy gave a name to microbiology. So originally it was this. And then some diseases happen from non-visible pathogens, like viruses. As you may know, and if you don't know, I will remind you because it's very important, viruses were first identified like so many other things in plants. TMV was the first one to be identified. And it was very difficult to identify them exactly because they were not microbiologically seen. You could see them in many important ways, and the most important ways that we consider pathogens are there is that we can somehow, in a controlled environment, reproduce what they are supposed to do as a disease. So you can isolate them and infect the next person, plant, animal, and get again infection in the same way you got it before. And viroid research it became something because some people, specific people that I will show you in a while, realized that there is an agent that produces a viral disease, but it didn't seem to be a viral agent. That's how it all happened. Now, because I may be talking for long and I shouldn't because I have to finish in less than 40 minutes, I would like to stress out here something that I consider very important. All pathogens need the same amount of genes, more or less, to do the job. This is something very important to remember when we start talking about the fact that viroids do not encode for any. That doesn't mean they don't need genes. They don't encode them themselves. They're smart enough to let others encode them. So no matter if you have, as a pathogen, 150 genes, 50 genes, 20 genes, 1 gene like hepatitis delta, or no genes like viroids, in the end of the day, you need a set of genes to do your job. So if you don't have it, you just hijack it. That's how it works. And this is the first, I think, concept why viroids are super fascinating. Because all the other pathogens, in contrast, look so stupid because they just have to encode for the proteins that do the dirty job, whereas viroids don't. They just steal everything. Okay? Now, stealing is one word, but in reality, host pathogen interaction, and pathogens here with inverted commas because they're not always pathogenic, is a much more complex story than it sounds. So usually from human nature and from human experience, we think that pathogens is a bad bug that we have inside us. Yeah, so my son, which is now ill with fever, actually has streptococcus. He says, why are these creatures needed on Earth? Yeah, it makes sense to ask this question. But the reality is much more complex. So none of the pathogens is there just to make somebody else suffer. The only, I think, pathogen that can do that is humans, that can make others suffer on purpose. Everybody else on Earth, everybody else, just do something that they will allow them to survive in evolution. So very, very, very often, I think we had this discussion yesterday with Manfred, or the day before yesterday with a beer, very, very often pathogens in nature don't want to be pathogens. And in fact, we know that from virology very well, that if you go out in nature, viral diseases of natural plants are much, much milder than they are in cultivated plants. I will not have time to explain why this is epidemiologically very important. And we knew this coming with COVID. We all knew this, especially plant virologists, because we have big experience with real experiments, not like animal biologists that cannot do these experiments. We knew that eventually viroid will become much, much milder to survive. That's a big discussion, a very interesting discussion. The reason I'm saying all that is because that viroids are not something that came there to steal from us. It came to share part of the biological material that gets energy from the sun. So to make it clear, very, very few natural existences on Earth live on their own. Phytoplanktons, maybe, yeah. Plants, so and so, because as you know, without microbes in the roots, they don't live. And then it gets worse. And then you get to huge parasites like us that we eat everybody else, literally everybody else, unless it's very poisonous, we eat them. And then on us, part of what we parasitize on others, others come to take part of it. So viroids are just part of this chain. They're nothing amazing in this sense. There's nothing particularly pathogenic about it. In fact, many of them are latent. As you know, all of you that work in grapes, they're full of viroids which are not very important. And as we heard here from my friend George, sometimes they can be also very helpful in cultivations. Not only they're not pathogenic, they are helpful. They may save the paradise's production of oranges. Okay, so first reason why you should be super proud to work with viroids is because they are evolutionary, seemingly the smartest of all pathogens, because unlike us, which are pathogens using 25,000 genes and an extremely complex brain to do what disastrous things we do, they do less disastrous things and they don't encode any gene. Yeah? And still they survive evolutionarily almost as well as we do, maybe even better, the way things go. So viroids are absolutely fantastic because they don't encode any protein. And what is absolutely amazing and is revolutionizing science every day, it's amazing how biologists don't realize that, because whenever I give a talk about virologists, you have this jaw-dropping effect. And they keep asking, do they not encode foreign proteins? Do they not encode? So how do they do it? Because structurally, if you think about it, they have to do everything by structure. Of course, structure is part of sequence as well. But this structure is also very tiny, so even the structure must be very simple. As you saw today, there was this very nice talk about this bending of the viroids, which is amazing. And this is as complicated as you get in terms of structure. You don't get something much more than that. Okay. I'll come back to the story, to the history, but I think that this is more important than the actual history. So, as I said, you have many different types of pathogens. In all my figures are stolen, borrowed, you would say. This is what humans do. But they're borrowed because I've put here the citation. So that's what happens inside. You steal something, then you cite it, and it's called cited material. So here I have the different types of subcellular pathogens. There are many. We're talking about viroids, which are of particular interest here. But as you saw in the previous days, and I thought it was very, very fascinating, I totally agree with Francisco on this one, I found the non-viroid RNA tiny pathogens extremely exciting for various reasons, not only because they make us feel that we're part of a normality of biology, but also because they are so, they show all the possibilities that RNA biology on its own has without the protein productivity. I mean, because even if you produce one gene, that doesn't solve your life problem, like in hepatitis delta. You have to use another 30 genes of the host somehow to be there. And then you use also genes of hepatitis beta, at least in humans. Okay, so the guy behind all this is this guy here, but I'll be talking about him in a while. So it was an interesting time in the States. I think George Vidalakis is the last of this line of people, because it was a time in the States before things went very wrong, that I think I will never get into the States again. That was my last visit. So it was a time when agronomical institutions in the countryside produced super science. You may have heard of Barbara McClintock. Barbara McClintock was, you haven't heard of Barbara McClintock? Wow. Discussing about the new generation of scientists. So Barbara McClintock, so maybe I should switch my talk to Barbara McClintock today. So Barbara McClintock is a fantastic person. Her biography, not autobiography, she would never write an autobiography. Her biography is out there, you can buy it. She's a Nobel laureate. One of the first women Nobel laureates, and definitely the first woman agronomist that received the Nobel Prize. She received the Nobel Prize for the transposable elements. But what is amazing about her, she did two amazing things. Well, she did many amazing things, but one amazing thing that she discovered transposons. The amazing thing is how she discovered them. And the second amazing thing that she did is that she discovered recombination. Recombination she discovered in a PNAS paper in 1932, at the same time with Max Delbruck in Drosophila. And the amazing thing with her, she was working in an agronomical institution in the States, yeah? Not something flashy, not MIT, not Harvard, not agronomical institution. And she was, how are they called? A karyologist. Karyologists were the people that would study chromosomes, but in the way chromosomes were thought in the 30s. So protein-based, not DNA-based. They didn't know, they thought DNA was just holding the proteins which were important together. So Barbara she was working with corn, as you would do, where she was working, and she had learned all the corn chromosomes and their zonations by heart. This is very important. So the chromosomes, they had some zones on them. Actually, chromosomes in Greek means bodies with colors. And this is because they got stained. And because they got stained, they could be seen. And because they could be seen, they could be somehow studied. And they had some zonations on them. So she could see the zonations under the microscope by eye. She could see the zonations, and she could see that the zonations would exchange after crossings. So you could tell that there is crossover, that there is a combination. But she didn't get the Nobel Prize that. She got it for transposable elements. So in the long line of extremely important scientists that came up of the agronomical institutes, there were some people in the States that work with viruses. That's how also they discovered viruses in the first place. So I should first jump to him and then come back. So one of these guys was Ted Diener. Ted Diener was working, he was a German scientist, seminal to the viroid research because he's, to us, the father of the viroid research, although George says that it's also semantic, which I totally agree. He was also important, but he nevertheless was first. Yeah, maybe they independently work at the same time. I agree on that as well. But practically, luckily, whatever, he was the first to publish. So this guy didn't work. We always say we celebrated 50 years of viroid research, and also the talk was named 50 years of viroid research. But it's not true. It's more than 50. So people started working with viroids when they identified symptoms of potato spindle tuber viroids or citrus barks, but they just didn't know the etiology. They didn't know the pathogen. So they studied them without being able to identify the actual pathogen. So this guy started working with viroids, and his report is a very interesting one. Already in 1967, there is a paper out from him that says that what he identifies as the etiological factor of potato spindle tuber disease, which he called the virus at the time, did not fit with the virus. And why did it not? Because technically, it did not isolate together with the viruses. So they would do a centrifugation, and the viruses, the viral particles would go to the bottom of the tube, but this was not the infectious part. The infectious part was at the top. At the time, he did not have the technical abilities, he got them later, and that's how he identified virus, to separate the agent. But he could immediately, already in 1967, realize that it's not the viral part, it's something else. Of course, there were other theories. He states them in his reviews. There are other theories why it was made, but it was broken parts, and they were not floating, going together with the viral particles. Maybe it was nucleic acid coming out of capsids but being infectious. Now we know that there are viruses that are not encapsidated and so on. But he worked for a long time in this research institute to find something so seminal. You could never do this research today. Nobody would ever pay you for 6 years to do in an agronomical institute basic nucleic acid toy play. Yeah, George, I guess. Yes. But at that time, they were, I would put it, they're more open-minded. People were more open-minded. The world was slower. So slower pace was accepted, was anticipated, was tolerated, and people had the time to think again and again and do the actual experiments to find out what is important. Where do I have my, come on. I need a slide that I have to find, so give me a second. That's the one I want to find. So this is from Diener's actually review. He goes back and tells us the experiments he did to identify the viroid. So first of all, as I said, he did a gross centrifugation. He realized that viral particles were not the infectious part. He realized that the supernatant was. Then slowly, he realized that this is a real pathogen because he could take an infected plant, and from the infected plant, he could do new infections. He could isolate the juice from the plant, separate it, and this part of the juice against was infectious. So he knew that he had infected agents that seemed to be a nucleic acid. That's what he said in 1967. And then he managed eventually to get a chromatogram and get a specific peak that fit to the specific, different peak than anything else, and that was infectious, and he could show that there was a specific particle in there that was doing the infection. That's why he was able to say that what he had in his hands was a viroid, okay? And that was this paper here. Now, of course, I wasn't there, I have no idea, and in fact, I was very maybe optimistic, and I didn't ask the people that have met Diener what did he think when he found his this amazing finding. So the only things I know are the readings I have from his own narrations. I don't know anything else, so maybe other people here that have met, maybe Ricardo, maybe discussed with you what Diener said about this. He definitely realized that it was something very important, and he realized it. We know this not only in retrospect, we know it because he started working with these bugs in 1965, and he continued for all over his life. So he realized that he had something very important in hand, and he started publishing about that. And at that time, as somebody very nicely said earlier, because the techniques were mature, many people at the same time identified viruses, started identifying viruses. But imagine, it's a totally different thing to have all the community against you, everybody to have the theory that viruses have a capsid, and inside the capsid is nucleic acid, and you need this, and you need genes, and these genes produce proteins that allow you to infect your plant, and try to fight against that. It's a totally different story to have established a paper that says that there is a non-coding nucleic acid subcellular pathogen in there, and now the community has accepted it, and then to say, oh, I've got also one in my lab, which is what happened. So we understand why then people started identifying other viruses. It was so much easier. But then things became very, very important. This is a timeline from one of the reviews. I may come back to this, but it's not so important for me. So I will skip that at the moment. So for me, it's extremely important, but it's extremely important, I'll tell you why, that they also managed to do an electromicrogram to actually see the viroid. The viroid is these little things here, these rod-shaped things here, and this is a T7 DNA. And probably it was for Diener important as well, because he has it on the back of his photograph. So I guess he considered it to be a very important photograph as well. And why is that important? Do you have any guesses, the PhD candidates? Why is this so important? It is very, very, very important because all these pictures you saw, with the exceptions of the one from my friend Ying, all the other pictures, or most of the other pictures that we show, including all the ones I showed, are 2D depictions of the viroid. But nothing in life, in real life, except of presentations exists in 2D. So this is, this is totally naive. This is what my son would have drawn 10 years ago if I asked him to draw a virus. It's not that somebody didn't do a good job. They did a fantastic job to simplify it, and so we understand what it means. But in reality, what happens is this. And this is super important because it's one thing to say that viroid is a rod shape and so on. Another thing is to see it. So this is a very important microgram because people saw it. And what is also extremely important, go back and front. Oh, this is Semanchik. That was the person that George was talking about. It was George's PhD supervisor. So only for this, he should have got a high price. Yeah. But nevertheless, he was also a founder of the viroids. It is claimed, I believe that he worked on viroids independently. But what we know is that he published them after Diener. So he was practically second, let's say. Okay, just don't worry. But that was just papers showing that the viroids are non-coding. So here is another fantastic microgram. And it's from 1976. It's from Senger's work with Riesner inside. And it's amazing because you see the actual nature of this smallish, medium-sized RNA, non-coding RNA. You see it rod-shaped. And then when you change the conditions, change the temperatures and so on, it becomes circular. I mean, that's fantastic. That's absolutely fantastic. Because now you can see it circular, which is amazing. I mean, I've never seen circularity so clear as here. And we don't, because usually we draw viroids as they are in the normal physiological conditions, which is rod shape. We always say, in all our introductions, including mine, we always say viroids are circular, linear, circular, non-coding RNA pathogens. But this is not exactly true. I think we should change this. I suggest to my more senior colleagues here that we stop calling them single-stranded. They're mainly partly double-stranded. That's what they are. They're only single-stranded when you boil them. If you don't boil them, they're mainly double-stranded. 8 out of 10 of their nucleotides are double-stranded, so they're not single-stranded molecules. They can be, of course, but this is true for every DNA and RNA. If you boil it, it becomes single-stranded. So I don't know why.
You have to say a single molecule with high conformation.
Well, absolutely, single molecule, but it's mostly found in a double-stranded form. That's what I want to say. Okay, so. By the way, all these people you see there, they're younger than me, but they're more experienced in viroids. So you are very welcome to have inputs in what I say. So I jumped through these. Okay. Now, the next very, very, very important thing was to find out how this thing is replicating. And I've been continuously being asked, and we saw also a very nice lecture about that today from Ying. I've been continuously asked who replicates viroids. So, of course, the obvious answer that could not have been given in the 70s because we didn't know that they existed would be RDRs. Plants have at least 4 RDRs, plus, RDR 4 plus, up to 6, 7, 8. So you would expect that an RNA is replicated by an RNA-dependent RNA polymerase. At the time, we didn't know they existed, which is a bit lucky because the only possibilities we had were DNA-dependent RNA polymerases. And there have been very nice papers in very primitive time with very little technology showing that it's Pol2, and one of the most convincing ones, and the first ones, was this one here showing that alpha-manatin, which is Pol2 blocking, was stopping viroid replication. Okay. Okay. I don't want to show this first. I would like to show the paper from Jose Antonio. So once we realized, once the first generation of scientists realized that this is a non-coding nucleic acid causing these diseases, it is the causing agent of all these non-understandable until then diseases, people start trying to understand the biology of these viroids. Once in one paper of mine, I had written the VIRP1 helps the viroid complete its life cycle, and the reviewer very correctly corrected the biological cycle, because it's not a life cycle, because it's not in an obvious way alive. Yeah, so it's a biological cycle. So people try to identify factors that are very important in viroids' life cycle. And there are the very obvious ones, but there are less obvious ones as well. As I told you at the beginning, all pathogens need a lot of help from their hosts, which of course doesn't do that voluntarily. And viroids are the best example of this because exactly they don't encode for any proteins of their own. So people started looking at this, and very often we stumbled upon things which were not obvious at all. A very good example is Pol2. With today's knowledge, my educated guess, which would be totally wrong, that is one of the RDRs that do the replication. It was not, it was Pol2, and it's very important it was Pol2. Then people started suggesting, even I think Ricardo's group was suggesting that there's a DNA ligase doing, an RNA ligase, doing the ligation of the viroid, and in fact in chloroplasts it's tRNA ligase, which I guess is an RNA ligase. But Jose Antonio very nicely showed that in post-P viroide, it's a DNA ligase that, again, like DNA polymerase, works to ligate RNAs. And in the lab where I was working at the time there, but I was not working with viroids, I was working with RNA silencing. Actually, we were collaborating in a program with Manfred at the time in RNA silencing, no relation with viroids. Emilio and some other people from the lab, Rudolf mainly, but not only, try to identify viroid interacting factors directly. So they do a system which I won't have time to explain, which is called a 3-hybrid system. It's like a 2-hybrid, but you need a byte for RNA. So it binds to RNA, and this activates something in yeast that gives a signal, and then you know that you have an activation. And with this method, which is a very bold method, and it's very bold because it took ages to do, and they were lucky enough to recover one important, but one protein. They recovered 2, but the other one proved not to be VIRP2, proved not to be important, have low affinity to viroids. But this was very important what Emilio did for various reasons, including the fact that he got his PhD with this, but also because it was a gene which was totally unknown. And for a very long time, as I told you until the first day of this meeting, we had also no idea why the plant would have this gene. This was the first gene that was identified with no obvious function inside the plant. So it wasn't an obvious gene already doing something we knew, like a polymerase that does a polymerization, a ligase that do a ligation, but here it was abducted. This is a gene that was not known to do anything else before. Okay, this I would also like to show because I want to pay also tribute to Martin Tabler and Biao Ding. They're both people that in my career had left an important effect for different reasons, and they both had very scientifically unhappy endings. I'll come back to that. It's not funny, this part. The rest is funny. In the same year, Martin, actually Yanis, then at the lab, master student, and Asuka Itaya in Biauding's lab in Ohio, they saw that RNA silencing, which was then a very, very new thing, and RNA silencing was identified practically in 1998, although it has a history before that. But very soon after, that's 2001. So in order to publish a paper in 2001, you started working with that at least 2 years before. So immediately after the finding of RNA silencing, it became obvious that a partly double-stranded RNA, which is naked, would be the first target for silencing. And it's still a miracle, let's say, that the viroid manages, with a naked, mostly a double-stranded RNA, to escape the silencing machinery of the plant. And originally, it was thought, there were experiments on that dicers are not able to digest that kind of double-stranded RNA. Now we know that dicer 1's ideal substrate is this kind of substrate. So would you expect it would destroy it, but it doesn't destroy it. So I think this is very important as well. And for a long time, you were not there, but my colleagues remember that, there was a very long debate if, and there still to some extent is, if silencing is something that the viroid uses on its own merit, or it's something that fights against viroids. I believe it's both. It's mainly a defense mechanism, but like all smart pathogens, it takes advantage of this mechanism on its own, for its own merit, like HIV does with our immune system, let's say. Okay. Did I forget anything from before? I did forget 2 important things, yeah. So I forgot, no, where is it? Come on. Where is the Nature paper? Sorry for that. Yeah. So there are 2 very important things I would like to say. How much time do I have until I finish? Perfect, perfect. So, viroid research has been extremely important for many findings. And as I said, you have to go back to the review by Steger in 2018, I think it is. Maybe even later, I can send it to you, where he exemplifies all the reasons why viroid research has been so important for other parts of molecular biology. Professor Riesner, the day before yesterday, also showed why it was important for the finding of these columns that we'll use for the separation of our plasmids. It was based on our viroid research. But also it's extremely seminal that in 1978, the complete structure of a viroid was found and sequenced. It's super important because at the time they had to find, it's not like today that you do a sequencing like that. It's not like even what it was 30 years ago where you could throw it in a machine and slowly you would get the sequences out. It had biochemically that it had to identify each and every single nucleotide and its position. And it's amazing that this sequence is 100% correct. No machine today produces 100% sequences. There's none, 0. This is 100% correct. Another thing which is first for viroids, and people don't remember that, maybe some do, but generally it's not very well established, is that viroids is the first system where RDDM, RNA-dependent DNA methylation, was identified. So it's the work of Michael Wassenegger, another sad story, by the way, as a personal case. Michael Wassenegger, an excellent scientist and a very smart person that at the time had identified in Sanger's lab, he had seen that when you infect plants that already contain viroid sequences in their genome, when you infect them with viroid, the DNA sequences get methylated. So something guides the methylases to the specific locations of the homologous sequences to methylate them. And this is 94, so it's, I think it's 94, yeah. So it's 94, so it's at least 7 years before RDDM in any other system was shown, which I think is quite amazing. Okay, and with this I'm almost finished. I would like to pay tribute to 3 people that you have not met. I will start with this gentleman here, and I will start with this gentleman here because this is Ricardo Flores, and he's giving a lecture at the Viroid meeting in Valencia 9 years ago. No, Marcos? It was in this room there.
I think it's
Maybe it was another meeting. Okay. Okay. So Ricardo, as you will, if you do a quick search on your Google, you'll realize that this has been seminal in most of the findings that we're discussing today in all parts of Viroid research. He made a lot of seminal findings, and amongst other things, he has left a lot of students which are guiding today the field. So all the part there you see are his students. So we are very grateful. Amongst other amazing things he had, we shared one thing together. We both loved history. So we were a very boring couple when we were together. Okay. I'll go to this gentleman here, Biao Ding. So Ricardo passed in a relatively, not old age, but later than the other guys, and quite recently. Now, this guy here, Biao Ding, he was very important, and you can see that he's very important because if you look at his publications, nobody continued his work. Maybe you're doing some of his work, a little bit. Because we all left the structural part that he was dealing with, which is very hard and very interesting and very, very, very important work. And he was the only one doing it, and we more or less went all to our comfort zones. I went to genetics, which I understand better, and the other guys went to other things, biochemistry and so on, and bioinformatics, which is all extremely important, but we left what Biao was doing, and now maybe Ying is continuing his work, I hope. But what is also important, coincidentally, with Biao is that Biao passed away the first day of the Viroid conference in Prague, which was in 2015. Literally, he had the first keynote speak, but he never made it. And this guy I left last because he was my supervisor. He was a person that put me in the Viroid field. It's Martin Tabler. Martin was a biochemist from Germany, Ludwigshafen, next to Mannheim. He was a chemist, and his dad was a chemist in BASF. This is important because Germans were the best chemists in the world for 150 years. So he comes out of a very big tradition of good chemists, and he looked at all the molecules as chemical molecules. He didn't see so much the life out of them, but he sees everything as chemical interactions. And for me, being an agronomist and then a biologist, this was extremely important because it gave me a new perspective of things. So with these three great people, I would like to leave you here. I once again want to welcome you. I had another slide that said, oh yeah, here it is, welcome, but I don't know why this is in the middle, doesn't matter. So I would like to welcome you. What it says behind is, welcome to the Viroid world, yeah. Okay, so I would like to once again welcome you here, and I'm very, very happy to continue the collaboration with you, and I hope you at least have inspired your enthusiasm in this fantastic little bug doing so many amazing things. So thanks very much.